Hi everyone, welcome back. This week we're going to be focusing in on chapter 4 of our text, where we're going to be looking at the process by which our cells are capable of taking glucose and producing ATP. Now remember, it's not only glucose, but that's going to be our main focus. We can also burn or metabolize our fatty acids as well as, and it's not as ideal, but our amino acids. When we looked at cell parts, remember the membrane-bound organelle found in the eukaryotic cell that's responsible for the production of ATP that acts as our powerhouse. That is the mitochondria. All right, so we're going to look today at the following stages: glycolysis, pyruvate oxidation, Krebs cycle, and electron transport chain, and look at the various regions of the cell in which these different chemical reactions take place. Cellular respiration, also known as cellular metabolism, is the oxidation of food to obtain energy. Now, we've talked about this before, right? That ultimately all of the energy that we obtain as omnivores comes from the sunlight, right? Whether it is that we're eating plants or whether you're eating both animal and plant byproducts, ultimately the energy is all coming from the sun. Right, now, we're not going to be covering plants and the process of photosynthesis, but just a quick crash course. The UV rays from the sun, they interact with the chloroplasts that are specific organelles found within plants. And the chloroplasts are responsible for then taking the UV energy that they've received and creating glucose. Right, and that glucose is ultimately what either the animals are then eating to then produce ATP, or omnivores such as us that are eating both animal and plant byproducts are receiving it from either source. Right, so even if you're eating a hamburger and eating that beef patty, well, that cow at some point was eating grass. That's how the cow was able to store up that key energy molecule of glucose in their body. Now, another thing that helps to balance the cycles between the plant and animal kingdoms is that the plants are producing oxygen. So they're taking up the carbon dioxide that we're producing through cellular respiration, and they're converting that into both glucose, which we can use to make energy, as well as oxygen, which we then use to power aerobic cellular respiration. So our main focus for today is going to be on glucose, but remember, there are other ways that we can obtain energy, specifically through fat breakdown as well as protein breakdown. And what are the macromolecules? Lipids, proteins, carbohydrates, and nucleic acids. Now, the reason that each of our cells, specifically the mitochondria, are working to create all of this energy is to create these molecules known as adenosine triphosphate. Right? I've mentioned ATP so far. This is what ATP stands for. Now, I want you to take a look here at this portion of the molecule. So we've got here a nucleotide base, adenine, which is attached to a ribose sugar, which is then attached to, in this case, 3 phosphates. But what if we only had 1 phosphate here? What does this molecule look a lot like? And if you said nucleotides, you were right. All right, specifically, ribonucleic acid, the adenine that makes up our ribonucleic acid, looks pretty similar to adenosine triphosphate. The only difference is that our ATP molecule has an additional 2 phosphates attached to that phosphate group. All right, and the reason that we have these phosphates attached here is because the phosphate bonds possess high energy. So when we break those phosphate bonds, we're capable of releasing that energy in the form of electrons and using that to power different processes within the body. So when you break the phosphate bonds here and you release that phosphate group, now you have those free electrons to use as energy. We now have an adenosine diphosphate molecule. All right, so when we're producing ATP molecules, what we're doing is we're recycling the ADP molecules that have essentially been used up, and we're just adding an additional phosphate on to that ADP molecule. All right, so ADP is adenosine diphosphate with 2 phosphates. Adenosine triphosphate is our energy molecule with 3. Energy can be released from any molecule that has a carbon-hydrogen bond, right? Because what is it that's actually creating these bonds? It's the electrons that are being shared in this case because these are covalent linkages. All right, so anytime we break the bonds that are holding together these organic molecules, we are able to release the electrons and thereby use them in the form of energy. All right, some examples here of molecules that contain carbon-hydrogen bonds: methane, which is your natural gas; glucose, which is going to be our main focus for today; and then also fatty acids. Right, we're going to see how you can break down your fatty acids and take the product of just simply cleaving off 2 carbons at a time and adding those to an intermediate that feeds into the Krebs cycle. Now, the problem with processed foods is that they contain mostly sugar and starch. Lots of carbon-hydrogen bonds, so that's great for energy, but ultimately, once we've then stored up the excess sugar as glycogen and all of those stores are maxed out, any excess that we have is going to be converted to and stored as fat. Another thing too with processing is that typically when we process grains, we end up losing a lot of the key vitamins, minerals, antioxidants, and even in some cases, the fiber content, right? That's the difference between your white bread and your brown bread. Now, with simple sugars, right, anytime you have an increase in that, it's going to cause rapid fluctuations. This also tricks your endocrine system because you end up not being satiated, which is essentially the feeling of being full. And you end up with just more cravings, right? This is why they say that sugar is addictive. It's important to ensure that you are going to be receiving all of the healthy fats, fiber that you need, because the fiber is what's going to fill you up ultimately. Now, during cellular respiration, as we're breaking down that glucose, converting it into a number of different intermediate molecules, there are going to be a series of redox reactions, right? And that's short form for reduction oxidation. During these reactions between 2 different molecules, electrons and hydrogen ions are going to be moved from one molecule to another. So with oxidation, we're looking at a reaction in which a molecule has lost electrons. Now remember, electrons exist in covalent linkages. Say you're talking about a carbon and a hydrogen being bound together. The electrons are what are holding those 2 atoms together. When we oxidize any compound, we are going to be losing electrons, and those electrons are going to be passed on to another molecule. All right? And once that other molecule receives those electrons, that molecule is said to be reduced. This one's an easy one to remember because reduced, just think if you're gaining negative charges, the overall charge would be reduced in that way. Okay, so with oxidization, you're losing electrons, and with reduction, that molecule is gaining electrons. Now, there's a great mnemonic, Leo the Lion says GR, that can help you to remember where electrons are coming from and going to. All right, so first off, with Leo, that stands for loss of electrons is oxidation. All right, and then with GR, gain of electrons is reduction. When it's oxidized, it's losing hydrogens and electrons. And then when it's being reduced, it's gaining electrons and thereby gaining the hydrogens. Now, our main focus for today is going to be looking at how glucose is metabolized, how we take that glucose monomer and we break it down through glycolysis, Krebs cycle, and electron transport chain to ultimately produce ATP. Now, I've been talking about all of our different macromolecules and the 3 specifically that we see on the nutrition label, right? Our lipids, our carbs, and then our proteins. Now, something we're going to add to that today is some other information that we get from the nutrition label, which is the caloric value. Well, a calorie is the energy needed to be able to raise the temperature of 1 gram of water by 1 degree Celsius. In order to do something like that, you would need to add energy into a system, right? Calories are a measure of the amount of energy that we're going to be able to obtain from these different macromolecules once we are metabolizing them in our own bodies. For 1 gram for each of the following macromolecules, as well, I've included alcohol to give you a bit of perspective here on those fruity alcoholic beverages. But with carbohydrates, protein, and fats, what caloric value comes from 1 gram of those macromolecules? So you'll see here carbohydrates and proteins Carbohydrates and proteins, you're receiving about 4 calories per gram, whereas fat, many more calories per gram of fat. Now, there are about 3,500 calories, to give you perspective here, in a pound of fat if you do the conversion there. Now, that works out to 3,500 calories in a pound of butter. Now, alcohol, I put this in here as well because there's about 7 calories per gram of alcohol. When you think about any kind of sweet alcoholic beverage, you've got not only the calories coming from the alcohol, but also from the sweetener that's been added to that. It's important for us to be aware of what it is that we're taking in. When you're younger, your body, for the most part, is able to keep up with whatever it is that you throw at it. But it is true, when you get older, your basal metabolic rate just slows down. And so without exercise and increasing your total metabolic rate, you really have to ensure that you're taking in a balanced, diet that's not going to be too heavy on one food group. Because, for example, look at your sugars, pro-inflammatory. Any excess that does not need to be stored up as glycogen would then be converted to fat. So it doesn't necessarily, if you're eating a low-fat diet, if it's high in sugar or refined carbohydrates, it's still going to ultimately be converted back to fat. Now, there's 2 types of cellular respiration that we're going to talk about today. The first one's aerobic respiration, and we're going to look at that, where we have oxygen and our cells are capable of using that oxygen to be able to produce a lot of ATP. In anaerobic conditions, or certain organisms that don't have membrane-bound organelles that are capable of utilizing that oxygen, they require anaerobic respiration to be able to produce ATP. We're going to take a look at that as well towards the end. One example of an organism that utilizes anaerobic respiration would be our bacteria. So the process of aerobic respiration not only requires oxygen, but glucose. Okay, anaerobic respiration can occur as long as glucose is there without the oxygen. But for aerobic respiration to occur, we need oxygen. And the whole process is to take this 6 carbon, remember your carbohydrate ratios, 1 to 2 to 1, it's following through there. To take your glucose molecule, combine it with oxygen, we then produce carbon dioxide, which we're going to breathe out ultimately, water, and a bunch of ATP. So a relatively clean burn, right? And then plants are able to take up that CO2 and they're able to produce more glucose and oxygen. So it's this perfect circle of life. So we're going to spend a lot of our time today focusing on aerobic cellular respiration, okay, whereby glucose molecule in the presence of oxygen is going to ultimately be converted into ATP. Now, the stages, as I mentioned at the beginning, are glycolysis, pyruvate oxidation, the Krebs cycle, and the electron transport chain. We'll be looking primarily at the starting and end products, okay, for both of these cycles, as well as looking at what's being input into the series of reactions and what is the output. And I've created charts that I have included along the way, but if you just print out one of those, fill that in for yourself, it'll give you a nice overview of all of the starting and end products. That way you can sort of organize in your thoughts where all of these different processes take place within the cell. So the first stage is known as glycolysis. Now, glycolysis does not require oxygen. You're probably thinking, okay, I thought you said we were looking at aerobic respiration. We are. With oxygen, we can then move to this next step. All right, without oxygen, we cannot pass from the end product of glycolysis, which is pyruvate. All right, so for aerobic respiration, we require oxygen in order for pyruvate to be oxidized within the mitochondria. Now, glycolysis, as you can see as it's illustrated here, occurs in the cytoplasm of the cell, right? And this is another reason why anaerobic bacteria are capable of utilizing glycolysis to produce their own energy, because they don't have membrane-bound organelles. Remember, the prokaryotes don't have membrane-bound organelles, so they don't have a mitochondria that's capable of producing all that ATP. So within their cytoplasm, they're going to be simply breaking down glucose, producing pyruvate, and there's a way that they can recycle that pyruvate. For us, because we are aerobic, we utilize that oxygen to then allow pyruvate to become oxidized within the mitochondria, which sets off another cascade of reactions with the ultimate production endpoint being more ATP. So stage 1 is glycolysis, and glycolysis occurs within the cytoplasm, as I just mentioned, and it does not require oxygen, right? So that makes it the main energy source for those prokaryotic organisms because they don't have mitochondria. They do not have any membrane-bound organelles. And so they are just going to simply be taking that glucose, breaking it down into pyruvate, producing a few net ATP, and continuing that cycle over and over and over again. As we're going to see a little bit later, that's a lot of work just to produce the amount of ATP that they're producing. To give you a big picture perspective, the net production of ATP here in glycolysis is about 2 ATP. We produce roughly 38 ATP throughout all of the different processes. So definitely aerobic respiration has its advantages for us. Now, there's a sequence of chemical reactions, as I mentioned before, a number of different steps that have been broken down into just a few that you can see here. But ultimately, we are taking a 6-carbon glucose molecule, and through different processes, phosphorylation, and then splitting it up into 2, we are producing 2 3-carbon pyruvate molecules. So your starting product is your 6-carbon glucose. That glucose is then split up into 2 3-carbon pyruvates. Now, along the way, to actually get this started, we require the input of ATP. And ATP, remember, is adenosine triphosphate. That's our energy molecule. When we break the bonds of these molecules, that energy in the form of electrons is then used to form ATP, right? That's known as phosphorylation. Now, phosphorylation is the process by which we will take our recycled ADP, the diphosphates, and add a phosphate to it to then produce ATP. All right, so when we use that term that's being phosphorylated, that means that we are adding a phosphate onto the ADP molecule to produce the ATP. Now, we have 2 different types of electron carriers. In their oxidized form, they exist as NAD+, and the other one is FAD. Right? When they are reduced, remember, they're going to be gaining electrons and hydrogens. The NAD+ becomes NADH, and the FAD is going to become FADH2. We're going to see that other molecule a little bit later. 2 different electron carrier molecules. Their whole purpose is to become reduced here in glycolysis, reduced in the Krebs cycle, and then ultimately oxidized later on so they can give off those electrons to our proton pumps. All right, so here in glycolysis, for each 3-carbon, we're going to have an NAD reduced, and we're going to have 2 ATP produced. So if you look at all of these totals, it looks like we're producing 4 ATP from glycolysis. But one thing you have to remember, to kick off the process of glycolysis, we require an input of 2 ATP for the phosphorylation to occur here. So if you consider the net then, you have 4 that are being produced, 2 is your input, so our total net would be 2 ATP. Okay, and we have that here. So you have 4 ATP output, 2 ATP input, and it gives you a total net from glycolysis of 2 ATP per glucose molecule. Now, glycolysis, as I mentioned, is the only way that an organism can derive energy from food in the absence of oxygen in anaerobic conditions. They must continue to just recycle their electron carriers to be able to then go back through the cycles, breaking down more glucose, producing that 2 net ATP. So here's a summary. You need to know the starting molecules. We're starting with glucose. Glucose has 6 carbons. How many ATP molecules are required for glycolysis to occur? That would be our 2. The number of ATP molecules made, that would be 4. That's the total. What is our net? Our net would be the 4 minus the 2 that we input. So that would be a net of 2 ATP molecules per glucose. How many NADH molecules did we form? 2. And then what is the finishing product? What was that final end molecule? That was the pyruvate with 3 carbons each. So you can see we've maintained the number of carbons in this case. All right, we've gone from a 6-carbon glucose to 2 3-carbon pyruvates, yielding 2 ATP and 2 NADH. Okay, so this is the chart I was mentioning. You can fill it in by yourself, or go to one of the last slides where I filled it in for you, and you can just print that off for your own reference. So glycolysis is taking place outside of the mitochondria. The rest of these reactions are going to take place inside, right, in the presence of oxygen. But it takes place in the cytoplasm. That's why any organism can use glycolysis to produce energy. We start with 1 glucose, we input 2 ATP, and we start with 2 oxidized NAD plus molecules, right? Those are the electron carriers. They need to be reduced. They need to take on those electrons. So what do we end up with? The 2 pyruvate molecules, 4 ATP total, 2 net, and then 2 NADH. And we're going to see where NADH and FADH2 come into play here as the starting products of the electron transport chain. All right, we're going to move on now to stage 2. All right, so glycolysis, we have produced our 2 NADH molecules, produced 2 net ATP, and now we are left with 2 3-carbon pyruvate molecules. Now, as I mentioned, in the presence of oxygen, pyruvate is able to then leave the cytoplasm and enter into the mitochondria, where then it goes through a process of oxidation. So it's here in the mitochondria now that 3-carbon pyruvate is going to be oxidized, which means it's going to lose electrons. So what ends up happening? Those electrons go off to another NAD plus to reduce it into NADH. And then your end product will be a molecule known as acetyl-CoA. Co stands for coenzyme, so you can also say acetyl coenzyme A. Right? And you might be thinking, okay, so we started with 3-carbon, and then we end up with a 2-carbon acetyl-CoA. Well, what's happening here? Well, this carbon gets bound to that oxygen, right? In the presence of oxygen, we can oxidize pyruvate. And so that carbon that's being lost because we've gotten rid of the bond there and we've given away the electrons, the carbon then can be bound to oxygen, which we then can get rid of and breathe out. Now remember, how many pyruvate are we starting with per glucose molecule? We are starting with 2 pyruvate per glucose. And so ultimately, we are going to be producing 2 CO2 molecules and 2 NADH molecules, as well as 2 acetyl-CoA that will each then enter into the Krebs cycle. Okay, so as I mentioned before, the specific enzyme that's going to be cleaving that bond there between the carbons is known as pyruvate dehydrogenase. Right? The single carbon is going to leave as part of that CO2 molecule. Those hydrogens as well as electrons are going to be removed from pyruvate, which is oxidation, and then they're going to be donated to the NAD+, reducing it into NADH. So now we're left with 2 acetyl-CoA compounds. So let's take these molecules and let's take a look where they're going to end up in the Krebs cycle. So now we're into the mitochondria. 2 pyruvate were oxidized. We also needed to start with 2 NAD+ molecules. We ended with 2 acetyl-CoA, 2 reduced NADH, and 2 molecules of carbon dioxide. Okay, so we are in aerobic respiration. We have broken down glucose into pyruvate. We've then oxidized pyruvate in the mitochondria, and now we've produced acetyl-CoA. So now that acetyl-CoA is going to enter into a cycle known as the Krebs cycle. Now you may have heard it referred to as the citric acid cycle in biology back in high school maybe. Krebs is just named after the individual that described it. Now ultimately, the fate of acetyl-CoA is going to depend on the availability of ATP in the cell. If there's not enough, acetyl-CoA is going to head off to the Krebs cycle. But if there's enough ATP, the acetyl-CoA is going to be diverted to fat synthesis for energy storage. So remember how I mentioned before, if we have excess sugar in our diet, once all the glycogen stores have been stored up and maxed out, then any of the excess glucose is going to be converted directly into fat. Now we're going to look a little bit later on at a process known as beta oxidation, where your fatty acids can be broken down into 2 carbon pairs, ultimately creating new acetyl-CoA molecules. So in that way, our fatty acids then, once acetyl-CoA has been stored up in our fats, we can also harness that energy storage to be able to feed directly into the Krebs cycle. Now, the Krebs cycle uses a series of 9 different chemical reactions whereby acetyl-CoA is then bound to the oxaloacetate, which is the 4-carbon byproduct of the Krebs cycle, to then form citrate, which is our 6-carbon starting molecule of the entire cycle. Now, citrate is also known as citric acid, and that's where this cycle also gets its name from. As I mentioned before, it can be either referred to as the Krebs cycle or the citric acid cycle. And so the Krebs cycle is this process by which this 6-carbon citrate molecule is then further oxidized while reducing our electron carriers and producing an ATP molecule so that all of these electron carriers can ultimately be sent off to the electron transport chain for the production of ATP. And it's a cycle. You start with one acetyl-CoA that's going to bind to the 4-carbon oxaloacetate, which is going to form your 6-carbon citrate. And then during this process, 2 carbons are going to be removed. And every time we remove a carbon in any of these cycles, remember, it'll bind to oxygen and be removed as carbon dioxide. So you can see one here during this reduction reaction of the NAD to form NADH. And then further along from 5 carbon down to the 4, you can see where another carbon dioxide is given off. At the same time, we are then using those electrons to donate them to NADH, which is being reduced. During this process of going from the 6 carbon to the 4 carbon, we have the phosphorylation of an ADP molecule to produce an ATP. So let's think about numbers here. We have one acetyl-CoA that's yielding 1, 2, 3 NADH molecules, and it's yielding 1 FADH2. And remember I mentioned this is the other electron carrier that we see. It comes into play a little bit later when we look at the electron transport chain. So for now, just understand there are 2 different electron carriers. In its oxidized form, it's known as FAD. And then once it's reduced, add those 2 hydrogens with the 2 electrons, it forms FADH2. Now this 4-carbon molecule is then recycled. You have the 2 redox reactions occurring. And then finally, this last form, known as oxaloacetate, then binds with another acetyl-CoA if there's a need for energy. If the cell's requirements are being met by the level of ATP in the cell, well then acetyl-CoA can be diverted to fat storage. However, if there's not enough ATP, then acetyl-CoA will continue to bind with the oxaloacetate, which will then lead us into another cycle. And remember, this cycle that's illustrated for you here, this is just a series of reactions that's taking place within the mitochondrial matrix. Okay, so just to recall here, glycolysis is taking place outside, and as soon as we're looking at pyruvate oxidation, Krebs cycle, and even electron transport, all of that's taking place within this region, the inner region of the mitochondria, known as the mitochondrial matrix. All right, so let's add them up. Here again, we are in the mitochondrial matrix. We've started with 2 acetyl-CoA, 6 NAD+. Let's go back here to this diagram. 1, 2, and 3, but remember, it's the 2 acetyl-CoA. All right, so we double up all of our numbers. So we've started with 6 NAD+ and 2 FAD molecules. And then ultimately, we're producing 2 ATP, 6 reduced NADH, 2 reduced FADH2, and 4 CO2 molecules. So now we're going to move on to looking at the electron transport chain. And this is where a lot of these end products, specifically the electron carriers, you're going to see all of them are going to act as our starting products for the electron transport chain. Outside of the mitochondria, what was that chemical process taking place where we took glucose, we created 2 ATP, as well as 2 NADH, and 2 pyruvate molecules? That was glycolysis. And so remember, we created pyruvate, and in the presence of oxygen, pyruvate is capable of then moving into the mitochondrial matrix, where it can be oxidized to form acetyl-CoA. Now, during this process, we have NADH being produced. That acetyl-CoA moves into the Krebs cycle, and through the Krebs cycle, we're producing 6 NADH, 2 FADH2, and then 4 CO2 molecules, as well as those 2 molecules of ATP. So this is everything right here that we've covered thus far with all of the end products. Now, remember, in glycolysis, we're producing 2 NADH molecules. And the whole reason that we're reducing those NADH is to carry those electrons onto the electron transport chain. Why? To produce ATP. So those NADH from glycolysis are going to have to pass in to the inner mitochondrial matrix, and then they will also play a role in donating their electrons. But they're a bit of an exception to a rule. So we'll talk about those after we've talked about the rest of them. So you'll notice here, embedded within the inner mitochondrial membrane, and to give you perspective, this would be your outer mitochondrial membrane, and the yellow would be marking your inner mitochondrial membrane. Okay, so all of these protein complexes we're going to be talking about, they are all located within the inner mitochondrial membrane. Now, each of these protein complexes serves an important role in helping us be able to create a concentration gradient within the intermembrane space. And we've talked about this before. We're creating a high concentration gradient and we're continuing to pump these hydrogen ions into this space. What type of transport mechanism must that be? And that would be your active transport mechanism. Specifically, these are the proton pumps that we were talking about before. These are membrane-bound proteins that are acting as pumps to actively pump protons into this intermembrane space. And we're getting close, and you'll see why we're doing this and creating this concentration gradient. So we have 3 of them, and they can be identified here. We're just numbering them protein complex 1, and if you want, you can add in there in brackets proton pump 1, proton pump 2, proton pump 3. Each of these has a unique role whereby it is going to be responsible for using the energy that's been stored up in these NADH and FADH2 molecules and using those electrons to activate the pump to send the hydrogen ions into the intermembrane space. Now, NADH and FADH2, they operate differently. Let's start with NADH. So NADH comes along and it is going to be oxidized again. Remember, this is its reduced form. It's carrying the electrons. Why is it carrying electrons? Well, because we need to use that energy at some point where it's going to be really useful in producing ATP. Here's how it works. So your NADH molecule encounters the first proton pump. NADH is oxidized back into NAD. It donates its electrons. Those electrons are going to then power the proton pump, allowing it to pump that proton into the intermembrane space. Great, you might be thinking. So we've got 1 proton across. Now we need to go and get the other 2 proton pumps working. So we have different coenzyme complexes. This one is actually CoQ10, you've probably heard of as the more common name. But it's known as ubiquinone. And here's another complex known as cytochrome C. Right, what you need to know is that these are electron transporters, and they are going to pick up the electrons from one proton pump and then transport them to the other proton pump. And so in this case here with ubiquinone, it's responsible for transporting the electrons from proton pump 1 to proton pump 2. All right, so this is where we get into it's literally a chain of proton pumps that are transporting electrons. What's the point? To then pump those protons into that intermembrane space. Okay, so NADH drops off the electrons at proton pump 1. You get the proton sent across. Then ubiquinone then is going to pick up those electrons and it's going to transport them over to proton pump 2. Well, that's going to then activate proton pump 2. There you go. You get your second proton across. Now, cytochrome C is then going to be responsible for picking up from proton pump 2 and transporting to proton pump 3. Well, now there you go. We've activated proton pump 3. Another proton is sent across. Now, so look at that. For NADH started at 1 And those electrons activated proton pump 1, 2, and 3. So you had 3 protons that were pumped across for every NADH molecule. All right, that's important to remember. Now, once these electrons end up here at the third proton pump, we need to do something with them. Well, we're able to use them in the addition to the oxygen that's present. Remember, this is aerobic respiration, along with the protons that exist in this mitochondrial matrix to create pure water. All right, so you might be thinking, okay, but we haven't produced any ATP, but we've fired a lot of protons into the intermembrane space. So now that we've created this concentration gradient with the protons building up that concentration within the intermembrane space, remember they're always going to want to find equilibrium. So if you let them, they're going to want to pass back into the mitochondrial matrix to be able to balance out the concentration between the two compartments. Well, we actually have these specific membrane proteins that are known as ATP synthase proteins. And they're there to allow for a way for the protons to be able to pass back into the mitochondrial matrix. And this passage of the protons moving from the intermembrane space back into the mitochondrial matrix is known as chemiosmosis. I keep talking about the electron transport chain and how this is, where you end up producing all the ATP, but the truth is, it's the process of chemiosmosis where we actually produce all of the ATP. The rest of the electron transport chain is about building up that concentration of protons in the intermembrane space. Once they're allowed to then pass back through into the mitochondrial matrix, we harness that movement and are able to then phosphorylate the ADP molecules with phosphates to be able to produce our ATP. So this process here for producing ATP is known as oxidative phosphorylation. And this is different than substrate level phosphorylation. You've seen this term, I think, already throughout. And what this is referring to here, substrate level, is referring to the fact that by reducing or oxidizing the different molecules throughout the different cycles, we are able to produce ATP through those processes. All right. Now, because it's the transformation of one substrate or molecule to another, it's referred to as substrate level phosphorylation. Okay. And in contrast here, at the ATP synthase, we have oxidative phosphorylation taking place. And this is where we produce a bulk of the ATP through our aerobic cellular respiration processes. So now that we've been able to walk through the process from glycolysis, pyruvate oxidation, Krebs cycle, electron transport, and chemiosmosis, I'd recommend go and take a look at the video links that I've included in the YouTube channel so that you can give yourself a visual to how dynamic this process is going to be, especially the process here through the electron transport when you're actually able to visualize, hey, these electrons, they're going to be carried by this ubiquinone over to number 2. And then cytochrome C will actually come and transport from 2 over to 3. It's helpful when you have the dynamic visual to be able to give you a better level of understanding. Now, as I've told you, the general rule is that for an NADH, it's going to drop off its electrons at the first proton pump. Therefore, it's going to pass through how many? 3. So for every NADH, we are going to produce 3 ATP molecules. All right, now with the FADH2, they drop off directly at ubiquinone. And ubiquinone picks it up and takes it directly to 2. All right, so because we're not starting at 1 and starting at 2 instead, then there's only going to be 2 ATP that are produced because we're going to have electrons sending across from the second pump and then from the third pump. So for every NADH, it produces 3 ATP, and every FADH2 molecule produces 2 ATP. Now, you're looking over here thinking, remember this NADH that was produced in glycolysis? Well, actually 2 from every glucose. What happened to them? Well, something a little bit different. It doesn't follow that rule. The NADH from glycolysis, they actually, you can see here, they bypass that first proton pump. And so they act similar to FADH2, whereby their electrons are given directly to ubiquinone, and then ubiquinone takes them to proton pump 2. So for the two NADH that are produced during glycolysis, they are only going to yield 2 ATP each. As a general rule of thumb, the NADH molecules produced through pyruvate oxidation, Krebs cycle, they are going to be producing 3 ATP. The NADH from glycolysis is only going to produce 2 ATP per molecule. And so if you work out the math, you'll see that we had 2 NADH here, 2 NADH here, and then 6 here. So it's 10 ADH and 2 FADH2 for a total of 32 ATP. All right, so let's take a look at our chart here for our final portion. We're looking at the electron transport chain, remember, taking place within the mitochondria, but now the 3 proton pumps as well as the ATP synthase are all embedded within the inner mitochondrial membrane. Now our starting products, we had 10 NADH. Now I've broken this up so that you can include this in your chart, where you have 8 that are coming from pyruvate oxidation as well as Krebs cycle, and then you have the 2 NADH that were coming from glycolysis. We also have 2 FADH2 as well as the oxygen needed to be able to produce the water as the end product, and as well as the oxygen that is capable of receiving those electrons at the very end of the electron transport chain and producing water. All right, so that gives us our end products of water as well as the 32 ATP. So I think you can appreciate by now, if all you were utilizing was the process of glycolysis to be able to produce 2 net ATP from 1 glucose molecule, you're going to be working pretty hard. And then you can compare that to our capabilities of being able to produce 38 ATP from 1 single glucose molecule because of aerobic respiration. And there we go. So if you're feeling tired, it could be that you are lacking in a number of the key vitamins that are responsible for the production of certain key players in this whole process. So for example, here, if you just take a look, a number of the B vitamins are responsible for producing different intermediates as well as the electron carriers that are so key and responsible for carrying electrons from the Krebs cycle to the electron transport chain. As well, coenzyme Q, also known as coenzyme Q10, CoQ10, may have heard of that. So that is your ubiquinone. And as well, your omega-3 fatty acids, because remember, all of the electron transport chain is taking place within the inner mitochondrial membrane. Now, we focused a lot today on looking at glucose and how glucose can then be utilized to produce these 38 ATP per molecule. But we know we can also utilize all of our other macromolecules, specifically the monomers, to be able to then break those down further and metabolize them to be able to make ATP as well. Okay, so quick review. We have our 4 different classes of macromolecules. Each of those is made up of their individual monomers. We have our polysaccharides, so our carbohydrates, our proteins, our fats, as well as our nucleic acids. And then remember, each of those macromolecules are made up of the individual monomers. So we have glucose that we focused on today. The amino acids we're going to take a look at, as well as fatty acids and how we can utilize those to be able to also produce ATP. And then as well, I know we've talked about how nucleic acids don't provide us with a lot of caloric value, which is why they're not included on the nutrition labels. But there are pathways through which we are capable of breaking down nucleotides further. It's just the least advantageous route for ATP production. All right, and so this is the mechanism by which we can enter into any of these different stages here using these different macromolecules. So today we focused on how a glucose can be fed through glycolysis, pyruvate oxidation, into the Krebs cycle, and then finally all of those electron carriers donating to the electron transport chain to produce ATP. Now, the second line, as soon as you've kind of used up all of the glucose stores, is that your body will start to burn fat. And so you can take your triglycerides and break them up into glycerol molecules as well as your fatty acids. Now, the glycerol is capable of feeding into glycolysis directly and then being able to pass through the entire process. And then with the fatty acids, we're able to take those carbon-hydrogen bonds within the long chains of fatty acids and break them up into pairs. And each of those two are used to make your acetyl-CoA. Right, so remember how acetyl-CoA was the 2-carbon byproduct from pyruvate oxidation? Well, if you take 2 carbons from your fatty acids and then you add on a coenzyme A to that, process known as beta oxidation, then we're able to create acetyl-CoA, which can then feed into the Krebs cycle. So you can see here, really all you're missing out in terms of ATP production is the 2 ATP from glycolysis. Beta oxidation is definitely an advantageous route for us to be able to produce ATP from another macromolecule. And then our third line, this is one of the least advantageous routes, would be breaking down amino acids. So it goes through a process of deamination. We are able to convert into different intermediates that can enter at multiple pathways throughout the different cycles. The biggest problem with breaking down proteins is that we end up producing nitrogenous wastes. Okay, I'm hoping you recall I made a big deal about this. We talked about how proteins and nucleic acids both contain nitrogen in them, right? So at the end of the day, when we're breaking down your glucose, what do we end up with? CO2 that we breathe out and water, right? It's a clean burn. Same thing with your fatty acids. When we're looking at proteins, nucleic acids, we have this leftover nitrogen and we've got to get rid of it somehow. And so what we do is we convert that into ammonia, so NH3. Now, ammonia can't just circulate from our liver. Once that ammonia has been produced, we then need to convert it directly into a product known as urea. And urea is capable of then being transported through our blood, eventually being filtered out in the kidneys, where we can then excrete it from our bodies through urination. But we have to get rid of it. And so that's one of the negative side effects of having to break down proteins as well as nucleic acids, is that the nitrogenous waste has to be dealt with. Now, if you think of somebody who's entering into starvation mode, well, the first thing the body's going to do is tackle those glycogen reserves and get rid of all the glucose stores. The next thing that the body's going to do is start to break down all the fats. Now, as soon as that happens, then the body's going to start to target the proteins. So think about this. You have proteins everywhere. One of the places you're going to see this, where the body's breaking down its own proteins, is in muscle wasting that's going to occur. So that's why typically when an individual enters into that mode, essentially where the body is just grasping at straws, trying to find energy from any source, you start to see the real, muscle wasting occurring because the body now starts breaking down its own proteins. So hopefully this gives you a really good picture for how our body sources and, deals with the sugar that we take in, helps store it, can convert it to fat, but we now know, hey, that's great because we can also then burn that fat later on. All right, so ATP yields. One NADH molecule will yield about 3 ATP. What was the exception? The NADH molecules from glycolysis, right? They will yield 2 ATP. So approximately from every glucose molecule, we are producing 38 ATP molecules, right? 32 from oxidative phosphorylation, 2 from the Krebs cycle, and another 2 from glycolysis. So if you take this ratio that you're making from a 6-carbon glucose molecule, making about 38 ATP, how many ATP molecules would we make from one fatty acid that was made up of 18 carbons? What do you think? Well, it works out to approximately 114 ATP molecules. All right, so we focused on aerobic respiration for pretty much all of this lecture today. Now, some organisms live in anaerobic environments where they lack oxygen. Organisms like bacteria simply do not have the luxury of being able to utilize oxygen because they do not have membrane-bound organelles. All right, so in the absence of being able to use oxygen to be able to harvest that full yield from aerobic respiration, we have to rely exclusively on glycolysis to produce the ATP. Right, so remember, with glycolysis, we are only going to be netting 2 ATP, and then we're also going to be reducing those 2 NAD plus to produce 2 reduced NADH. Now, if we just kept going through this process and we had nowhere for these NADH to then be further oxidized again, we'd be stuck. And so anaerobic respiration is a process by which where the anaerobic bacteria are capable, through a process known as fermentation, to then have those hydrogen atoms as well as electrons from the NADH molecule be donated again to organic molecules to then regenerate that NAD+. This process of 2-pyruvate being converted to 2-acetaldehyde, one of the carbons then is given off in the form of CO2. And so this allows for the continuous recycling of NAD+, and this is the key point here. We're able to produce pyruvate through the process of glycolysis, but then able to convert that into an organic molecule, acetaldehyde, which can then become reduced itself as it oxidizes NAD. All right, so if we're reducing this, we're giving the hydrogens and giving the electrons to that molecule to produce ethanol. All right, so what we're doing is recycling the NAD to allow this process to continue. All right, well, this process of fermentation, this is how all alcohol is made. This is how beer is made. So an example here with yeast, which are a unicellular fungi. Remember that exception to the fungi kingdom. They're typically eukaryotic multicellular. The yeast are unicellular. In this case, pyruvate is converted into acetaldehyde, which then accepts that hydrogen from the NADH, producing the oxidized NAD+ and ethanol. Now, in animals, it's a little bit different in terms of the byproduct. We're able to, in anaerobic conditions, be able to convert that pyruvate directly into lactate, which is also known as lactic acid. And this is what occurs within our muscle cells. Okay, this is the burning sensation that you feel if you've been working out and you're working in a situation where you have an oxygen debt. As we run out of oxygen and we don't have enough to make ATP through aerobic methods, then we are going to be relying on glycolysis to be able to produce that ATP. And then the byproduct is going to be that lactic acid buildup in our muscles. Now, anaerobic respiration is actually used to ferment lots of different foods, all the way from yogurt, soy sauce, sauerkraut, kimchi, you name it. So we talked a little bit at the beginning about your basal metabolic rate, which just think of that as sort of you wake up in the morning, your eyes are open, you really haven't done anything, you're not moving, you're not eating, you're not exercising, okay? Your metabolic rate at that point, that would be your basal metabolic rate. So that's just the rate at which your body is going to use any of the energy stores in order to produce ATP for just your body to survive. Now, there's a number of things that we can do to increase our total metabolic rate. And one of the big ones is exercise, right? You're increasing the demand that your body is going to have for energy to be able to produce all of those muscle contractions. Exercise is also going to cause growth hormone production from the anterior pituitary gland. As you're stimulating those muscles and you're putting demands on those muscles, and now the more muscle that you're building, the more muscle cells that you have, they are going to require the most energy just to function. And so the more muscle that you have, the more ATP that's going to be required just at a basal rate. The more muscle that you build actually does pay dividends because it's going to be working for you when you're not working, essentially. All right, so having more muscle than fat also increases ATP requirements. Exercise also stimulates thyroid hormone production. Remember, thyroid hormones are your metabolic hormones. So if you have elevated levels of thyroid, your metabolic rate is also going to be increased. Now, another way that you can help to lose weight is just decreasing the calories that you're taking in. One of the most important things is to watch the amount of sugar that you're taking in. Lots of sugar is bad for so many reasons. Not only is it just going to be converted into fat if you have excess stores of it, but it's also pro-inflammatory. Lots of different conditions that occur with the body are a result of just high levels of sugar in our day-to-day diet. Now, minimizing starchy carbs to about 2 to 3 servings a day, and it's best to eat those after you exercise because then it's an easy fuel source. So when your body is wanting to repair the muscle tissue after your workout, it has an easy fuel source that's readily accessible for your body. Now, lastly, incorporating fasting into your weekly routine. Now, I'm not going to hold you to any specific guidelines, but I want to be able to introduce you to some of the ideas behind why you would fast. Number one, it forces your cells to use stored fat. So think about this. If you continue just to give your body glucose all the time, it's just going to keep up storing the excess as glycogen. And then if you try to, watch what you're eating, Your body's first going to have to break down all of that glycogen before it thinks, yeah, now I'm going to go to those fat stores to go and produce energy. So by fasting, you're forcing your body to have to eliminate those glycogen stores, and then it has to then start to target the fat to start burning that. Also, when not exercising, fasting can help to increase growth hormone as well. All right. So, when exercising, you need to make sure you have adequate fuel, but when not, to go through a period of fasting can help to also stimulate growth hormone. And then also decreases insulin resistance. So, we're going to get to talking about insulin and the role that it plays in helping manage your blood sugar levels. And we'll do that when we look at the endocrine system next semester. But ultimately, diabetes, specifically type 2 diabetes, is caused because your cells have become resistant to insulin. And typically, that's due to the bad fats in our diet. And so the cells are resistant to insulin. Insulin can't bind to the receptors and can no longer then allow for the glucose to be taken into the cells as readily. All right, well, hopefully that was some food for thought. That brings us to the end of this lecture, but more importantly, to the end of the new material that will be on your midterm exam. Now, if you have questions in the meantime, feel free, as always, to send me an email. I'll get back to you as soon as possible. Otherwise, I look forward to seeing you during our tutorial. Take care.